Let's go to the next system. That would be citric acid cycle, i.e. Krebs cycle. Guess what? Your book calls it Krebs cycle, so I'm just going to go ahead and, I can accept Krebs cycle. I'm sorry, your book calls it citric acid cycle, but I'll take either one. I don't really care. Does that make sense? As long as you get it right. Don't call it something else that we learn next week. All right, so your citric acid cycle takes place in the matrix, and I'm going to draw it like this because it's a cycle. Boom. And it cycles around 2 times. 2 times. Boom. Okay? And the reason why it's a cycle is because there's a molecule that is just sitting waiting around in the beginning. Same molecule. You'll expect it. And then when you have your acetyl coenzyme A come in, 2 acetyl coenzyme A's will come in. Right before they come in, they'll actually drop off their enzyme A, their coenzyme A. But I'm not going to test you on that piece, okay? So they will come in, and this molecule sitting and waiting, ready to receive it, and then it will incorporate it into its molecule. Then you have a molecular change, and you have a new molecule. And then you go through a series of inputs, molecular change, outputs, molecular change, inputs, outputs, molecular change, all throughout. And the molecules are the same at every single stage, every single time. That's why they call it a cycle. Now, I won't make you remember those molecules that are in each of the stages. I'm only going to make you remember the inputs and the outputs. Sound good? When you get into micro, you're probably going to need to know about 7 of those molecules at every stage. That's why I'm just kind of getting you in there right now. Alrighty, and I'm not gonna hold you to where it comes in, where it goes out. I'm just gonna hold you to the total. So, what do we got going on total? We have, before you commit to this, if you're writing a pen, don't write this down yet. Okay, let me finish it and then I'll tell you when you can write it down. You have 3 NAD pluses going in, coming out. The reason why they go in is they're going to take electrons from this molecule, high energy molecule, because it's still carrying high energy electrons, because it was made from pyruvates and it had high energy electrons, and pyruvates were made from the glucose, and the glucose has high energy electrons. So this molecule still has a lot of potential energy that the cells need to extract from it. That's basically what's going on. So your 3 NAD pluses are going to go in. They actually go in at different parts, but like I said, I'm not going to test you on that. And they're going to come out as 3 NADHs. Don't write this down with pen until I tell you when. All right, so you also have coming out are going to be 2 CO2s. Boom, 2 CO2s. Then you have 1 ATP coming out, high energy molecule via, same thing as this ATP, substrate level phosphorylation. I'm just going to write sub-level phos. Enzyme is in there and that helps make it. You have 1 FAD molecule, oops, 1 FAD going in. This is another vitamin. It's another type of a B vitamin. This is a B vitamin also. That it's just a slightly different molecular configuration. It goes in, comes out, it's going to grab 2 H's. 1 FADH2. That is the big picture. But here's the deal. We got 2 acetyl coenzymes. We're going to cycle around twice. 1 acetyl coenzyme goes in, makes all of this. There's 1 cycle. The second acetyl coenzyme goes in, makes it also. That's 2 rounds. Well, there's 6 total if you count them both. There's 4 total. There's 2 total. There are 2 total. Make sense? So you make 3 of that per cycle, 2 of that per cycle, and by the way, different sources will change that on you. If you watch YouTube videos, different sources will change it. They're still figuring things out. So I'm going to go with what your book tells you. 1 ATP per round, and then 1 FADH2 per round. So FADH2 stole electrons from the molecules that came in, high energy electrons, So this also is going to get sent to the electron transport chain. High energy electrons dropped off at the electron transport chain, ETC. This gets sent to electron transport chain also. So the whole point of all of this is to make these temporary high energy molecules. And the temporary high energy molecules are 2 NADHs from glycolysis, 2 NADHs from pyruvate oxidation, 6 NADHs from citric acid cycle, so which means a total of 10 NADHs from all of those systems got sent there. And you get 2 FADH2s from citric acid cycle, also goes to the electron transport chain. I'm gonna write that one. See, that's why I always do my Christe in the wrong place, no matter what. It's always in the way. Back it up more here. There we go. There you go. Your FADH2, bless you, goes here. 2 FADH2. These are high energy molecules. These high energy molecules are going to be sent to oxidative phosphorylation, which is a combination of electron transport chain and chemiosmosis. And this one right here is the best guy of all because he's going to make the most ATP. So far, we've made 4, but really you only made 2 net, right? And then over here, you didn't make any ATP here. Please know that for your test. And you made 2 ATP here. So at this point, you've only made 4 ATP. That's not enough energy. That's just really not enough energy. So we're gonna go ahead and talk about the next system, oxidative phosphorylation.